He completely switched from wanting to be a medical doctor to just loving research. And so he's now a researcher at MSU, and I'm very, very proud of him. Welcome, Trent.
Thank you, Kate, for that introduction. I wish I could take you everywhere to introduce me. But unfortunately, a lot of the times the lab can only fund me. So I'm going to be here today just to kind of talk through my journey in science and how the Phages program really made a cornerstone for me in deciding that I want to go into science and pursue a PhD. So as like most of our lives, our journeys are not a straight path. It's always twisty and turny, and some of those turns are for good and some of those are not for good. But the Phages program was probably one of the best turns I've ever had in my life. Kate has been an amazing mentor that I've had in my life in shaping me as a scientist, shaping me as a leader by giving me opportunities with the HOSA program. And this is where everything just started. This is where I met Kate. In my senior year of high school, I was part of her Discover Biology cohort, where we got to participate in all the good stuff that we've been talking about this whole conference. So I won't go into all the details of what we did, because I'm sure Kate did an amazing job of explaining all that to you. So the next step that I took in my life was I applied to go to MSU as an undergrad. I got in, fall semester was great. And then in case if you didn't know what happened in the spring of 2020, there was a global pandemic and we were all told, go home. You cannot be here anymore. So we all got shipped home. We had to switch over to WebEx learning. Whole different change of pace in my freshman year where I'm just trying to grasp and understand how to do college. But we got back and there was a diagnostic COVID lab ongoing on campus. And I was like, whoa, this is kind of cool. And fortunately enough for me, my academic advisor at the time, Dr. Seth Walk, right over there, was one of the co-directors of the lab. And so I met with him to plan for my sophomore year classes and was like, hey, I saw you're running the COVID lab. Like, do you have any undergrad positions? Like, are you open? He was like, yeah, like we really need people to come and help like process samples and get it set up for the RT-qPCR stuff. The other co-director of that was Dr. Deborah Kyle. And as you can see behind her name, she has so many certifications. So she kind of served as our clinical liaison and got us our CLIA certification so we could set up the lab, effectively process samples for students and folks around the state. And then Dr. Walk ran a lot of the molecular biology side of stuff with the RT-qPCR. So he got our apparatus set up, he got our grants from the NIH to fund the projects, to get all of our machines and equipment. Yeah, it was a great time. And then towards the end of this semester, Kate emailed me and was like, hey, you remember that cool thing that you got to do with me in Anaconda? You can actually apply to do a summer research program if you want. And I was like, wow, this is really cool. So I went to Seth with this and was like, hey, if I get my own funding through SEPA and IMBRI, could I like transition from the COVID lab into the research lab with you? He's like, yeah, that'd be awesome. So I did that. In 2021, I did the 10-week program at MSU. And Seth Walk introduced me to two other amazing folks of his lab, Drs. Chen Wan and Rajenbeck. I call these my lab moms. They really took me under their wing. They're both experts in arsenic toxicology. Chen does a lot of stuff on the microbial side. And Dr. Rojenbeck, her PhD was all focused on like the physiology of arsenic and how it gets into our cells. So learning from these two folks was really instrumental. And at the time, Dr. Rojenbeck, like right as soon as I transitioned from COVID lab into the research lab, she just published this paper in the Journal of Toxicology showing that antibiotic perturbation of the marine, which are mice, gut microbiome introduces an inter-individual susceptibility to arsenic. So I'll kind of break this down really quickly to explain the importance of this. So what is arsenic? As I was mentioned last night, arsenic is a naturally occurring element in the Earth's crust. And because of this, it's found all over across the globe. It doesn't matter where you are, there's high estimates as shown here in red, all over across the globe. And a close example of this is my hometown of Anaconda, where you have like the slag pile that's now covered in grass, which is really weird and confusing to me. But there's a large amount of arsenic in areas like this due to mining activities. And as we talked about last night, this is like an insane amount of mining tailings that we just can't deal with it other than pick it up and put it somewhere else that we don't want to see it anymore. But if you go to the global scale and just take the average of how many people live in these areas, it's about 100 million people worldwide are exposed to unsafe levels of arsenic that contaminate their drinking water sources. And why is this a concern? Well, that's because arsenic is a very potent toxin and group 1 carcinogen in its inorganic form. Specifically, it'll cause cancer in the skin, lung, and urinary bladder in humans. And something else that's really interesting about this is there's this fascinating inter-individual variability to arsenic in the human populations that get exposed. A good example of this is an unfortunate poisoning event that happened in Wakayama City of Japan in 1998, where I think it was, 63 people were exposed. 4 of them passed away, and the others had some sort of illness. And the people that became ill but survived have really strong differences in what happened to them following the exposure event. Some of them had like really bad skin lesions for like the rest of their life, which is a common thing that happens after arsenic exposure, whereas others just had diarrhea the next day and nothing else. So it's really fascinating, and when you take arsenic exposure and try to put it into animal models like mice, you don't see any of this inter-individual variability. Really, you only see all the mice do really good or all the mice do really bad. So it's really fascinating that we haven't been able to recapitulate this in mice until our lab. And so going back, we have a bunch of people across the globe exposed to arsenic. So our kind of like, why is this important? And what we tell the NIH to get money is, in one hand, we have high levels of inorganic arsenic in our drinking water. And if you look at the data that the CDC collects on antibiotic usage in America, and you go and look at their data from 2017 to 2021, and average that, you get about 7 in 10 people in the United States take at least one antibiotic in an outpatient format. So saying, oh, I have an infection or I got an STI, they prescribe you an antibiotic and you're sent home. So this number doesn't even account for people in nursing homes or the ICU, where we know they commonly get an antibiotic. So on the other hand, we have a high rate of antibiotic use. And taking these together, our lab is like, well, maybe this high rate of antibiotic use and these high levels of inorganic arsenic are related, and maybe explaining why we see this inter-individual variability. And a lot of this comes from previous work by others in the gut microbiome research field, where we know antibiotics really kill our gut microbiome. Where before antibiotic use, we have a healthy plethora of microbes, so a lot of different types of bugs present there. You take an antibiotic and it's going to broadly wipe a lot of them out. And the antibiotic does its job that's intended for, which is to clear bacterial infections. But a consequence of that is it also is going to clear out some of your gut microbiome. And for arsenic exposure, a previous PhD student in the Walk Lab, Dr. Mike Coriel, had this really cool Nature paper, which if you're interested, I suggest go reading, showing that the gut microbiome is required for full protection against arsenic toxicity. Whereas we took like germ-free mice, which I'll explain what those are in a minute, but basically mice with no microbes living in or around them, really were susceptible to arsenic toxicity compared to mice that have a natural microbiome. And so this kind of led our lab to start questioning, well, how do antibiotics influence arsenic toxicity if we know they're going to wipe out the gut microbiome, and we know that the gut microbiome is really important for arsenic toxicity? And so to test this, we just developed a really easy mouse model where we just take some mice, give them an antibiotic in their drinking water, and then co-expose them to the antibiotic and inorganic arsenic. And then we continue this exposure for 28 days and just ask the question, what happens? And when Dr. Rogenbeck first did this in her publication, she found if you take just the antibiotic, 100% of the mice survive, which is good and what we want. You take only arsenic at the same dose that we're giving it in all the other treatment groups, 100% of them survive. You take a sham, which is just taking the antibiotic, heat denaturing it, so autoclaving it before we expose them to the mice. 100% of them survive. But if we take the intact antibiotic, do the co-exposure model that I just showed you, and run it, we get this inter-individual variability, whereas about 45% of the mice survive, and the remaining mice get sick and have to be euthanized. And when I say like sick and very different, like the healthy mice that survive never present with any of the symptoms that we track for, whereas the sick mice will start to get category 2 or category 1, and start to get mild to moderate symptoms where we start to have to come in twice a day and check on them, and then eventually reach grounds for euthanasia. These sick mice never reach that. So really strongly recapitulating what we're seeing in humans. And during my time in IMBRI, Dr. Rosenbeck and I ran this model over and over again, just to make sure that, okay, it wasn't a fluke, like this is actually happening, right? And so the first thing that we did is we just took weight tracking, because that's one of the things that we track for in these mice. And we found at the start, both sick and healthy mice rapidly lose weight, which we expect when we give them an antibiotic and arsenic. So we're really double whamming them. But as the experiment goes through, these healthy mice start to make a recovery back to baseline, whereas these sick mice just reach the grounds for euthanasia and can't continue on any further. So really interesting, and that's some of the work that I was able to do during my SEPA and IMBRI project. And then after this, I went into, or towards the end of my SEPA IMBRI project, I went to Dr. Walk. I was like, can you please keep me? And he was like, actually, we really want to keep you. Do you want to have an undergrad position in my lab where I just pay you off our research grants? We have plenty of money for you. And I was like, yeah, I would love that. So a lot of the work that I did here was running our germ-free mouse facility. So the Walk Lab in the ARC at MSU has a facility where we have what we call bubble mice. And so they live in these vinyl isolators and are completely sterile environments. So they're never exposed to any sort of microbe their entire life. They're a really powerful tool for microbiology research, because then we can completely remove the gut microbiome as a variable and study what is going on. And this is what it looks like closer, really generic name, but we call this Helga, our breeding isolator, because it's massive. As you can see, 2 people can fit at a time. And these are what the germ-free mice look like. They look like normal mice. We just have some broth down there, just make sure that we aren't able to grow anything, make sure the food's sterile and everything, just kind of routine. And then these smaller ones are our experimental isolators where we conduct our germ-free experiments. So I've done a lot of collaborative projects in the Walk Lab and led some of my own germ-free mouse studies during my undergrad. And from this, I got 2 published co-author studies with our collaborators at Penn State University. And with our collaborators out of Houston, I am actually now in the process of writing my first author manuscript. But all this work was done while I was an undergrad, which would not have happened if I didn't get into that SEPA IMBRI program with Dr. Walk and had it with the Phages project, thanks to Kate. So I'm just going to tell you a little bit about my first author publication. So what we did here is we developed a brand new mouse model to test antibiotics. So as I showed you with even our model, the most common way to test antibiotics in mice is just to put it in drinking water. But a lot of the antibiotics that are used in clinical studies are not water soluble. So some people have come around this of oh, we'll just put DMSO in the water. That'll make it where it's water soluble now. It'll be fine. It still precipitates out, so you're not getting a normal dose or a human relevant exposure. So what we've done is we decided, okay, we're really going to make this relevant to humans, and we're going to take our germ-free mice and give them a human-derived FMT. I'll let you put that together, a fecal microbiota transplant. So I think you got the picture of what I did there. And then I let them set up in a biosafety cabinet for a little bit. And then I switched their chow from the pellet style to powdered style. So I can take the antibiotic at a clinical or pharmaceutical grade that's used in humans, crush it up, and then able to expose the mice through the chow at a human equivalent dose. So to make it a lot more translational and clinically relevant. And then we did this with a bunch of different antibiotics used to treat C. diff. for time's sake, because I know we're all hungry and want to get to lunch, I'll just kind of skip through it. But the work that I did with this allowed me an invitation to go to CLOS Path, which is a big conference for Clostrides difficile researchers. And we got to go to Banff, Alberta, Canada last September. This is Kevin Gary here. He's our collaborator out of Houston, and this is his lab. They called me their adopted PhD student for the conference. It was really fun. And I got to talk to experts in the gut microbiome field, like Dr. Tor Savage here. It was a really fun conference and a really unique experience that wouldn't have been possible if it weren't for the Phages program that kickstarted all this. Also, a side note, if you haven't ever been to Banff, I highly recommend it. It is gorgeous, and I hate to say it, but it kind of puts Glacier to shame a little bit. It is this jaw-dropping beautiful. Like, we even had Northern Lights, like our first night there. So of course, my little science nerd self woke up at 2:30 in the morning, even though I had a poster symposium at 8 a.m. the next day to go and take pictures of the Northern Lights, because I have never seen them. So highly recommend that. So that was my undergrad, and then towards my undergrad, I was like, well, I don't really want to go to medical school anymore. Like, I'm like falling in love with this. So it was the summer before my last semester, and I texted my boss, or Seth. I was like, hey, can we like grab lunch? I want to talk to you about grad school. And I got like a reply like that from him, like, yeah, let's go to Bridger, which is a brewery down the street from campus. He was like, I'll take you out. And so we're like chatting, and he was like, well, do you want to do like a master's degree? And I was like, no, you know me, I'm going to do the full thing. Just jump to the end of the story. Let's get a PhD and do this. So the main project is building off of what I did during my SEPA Embry internship, building off of Mike Coriel and Barb Rogenbeck's work out of our lab. So kind of the main hitter of this is currently microbiome status and antibiotic use are not implemented into the risk assessment of arsenic. And as I've shown you with work that Dr. Rogenbeck and I have done, antibiotic co-exposure with arsenic increases its toxicity by about 4-fold. So this is a huge increase in the toxicity that's not currently assessed in those safe limits that we put on drinking water. So my project aims to further evaluate the influence of antibiotics on acute arsenic toxicity, and also broadening this out to some cancer models to see how that affects it as well. Just really briefly, I'll just show some of the prelim stuff that I've done as a grad student. So when Dr. Rogenbeck first did this, we had an in-house conventional mouse colony. And so when she did it, she got that 45% inter-individual variability. We no longer have this colony. Thanks to COVID, we had to eliminate that because we had to reduce our staff at the ARC, so we couldn't keep the husbandry line going. So we then had to transition to purchasing mice from commercial vendors like Jackson and Taconic Laboratories. And when we purchased mice from Jackson, we thought we lost the phenotype because only one of the 40 mice that we used survived the model. So we were like, maybe it was a fluke. So then we purchased mice from Taconic and ran it in the model and saw that they still had this inter-individual susceptibility that we saw with our in-house colony originally. So we went to the literature and we're like, well, these mice are genetically the same and inbred. How are they different? Well, their gut microbiomes are very different. So I've done a lot of work with the fecal samples that we collect throughout the experiment. And I've shown from the fecal samples that we collect at baseline, so before the experiment starts, extract DNA from those, send them out for 16S rRNA sequencing, and then get the results back and run it through our pipeline that we have in our lab to analyze it. And so what this graph is showing, if you aren't familiar with it, these are NMDS plots. And so each dot is representing one unique sample, so one microbiome of a mouse. And basically you interpret this graph as it is like a map. So the closer the dots are, the closer they are. So like, let's say this is Montana and this is Texas. So the closer they are, the more related and more similar they are. And so what I've done is I've just colorized them based off of which vendor they came from, and then gave them a shape if they were sick or healthy. And we didn't see any differences between sick or healthy mice, but we did see a huge difference between the 3 vendors of mice that we were using. So this has given us some implication that the crosstalk that we know of between the gut microbiome and our immune system is really driving this model outcome of whether you're sick or healthy. So we're hypothesizing that different microbiomes at that baseline from those different vendors are establishing different baseline immune responses that are driving this model outcome. I didn't show the data, but we have really strong evidence that our sick mice are actually succumbing to a sepsis-like disease state. So this is where we get this idea of the crosstalk between the gut microbiome and the immune system is really driving that difference. I won't go into the weeds too much because I know I'm kind of running out of time and over it, but basically one of the experiments I'll be doing this summer is testing this hypothesis with some of our germ-free mice by taking FMTs from those healthy taconic and sick Jackson mice and grafting them, reconventionalizing the mice and running them through the model just as a proof of concept that I can establish that same phenotype with those engraftments. And then lastly, the other thing that I'll be doing is broadening out our antibiotics. So currently we've only really tested cefoperazone, which is no longer in clinical use in the United States. It's used as like a last resort in Europe in very, very rare cases. So I'm gonna broaden out and take the top 5 prescribed antibiotics according to the CDC and run them through our model to see if we get similar effects or not. And so I've taken you through the journey that I've been able to have in science thanks to the Phages program and the CEPA-IMBRI summer research program that I did. And with that, there's a ton of people I can thank for this, especially the research team that I have at MSU. But really, I want to thank all of you in this room, especially the teachers who've taken rural kids like myself and put us in a classroom and show us the beauty of science and research. We may not all turn out to be scientists, but at least we can build a better society with more science literacy. So, thank you. If there's any questions, I'll take them.